How's it going, guys? Today we're going to be talking about the human genome, which is going to conclude with two different topics. One is going to be karyotyping, and the other is going to be mutation. Both of these topics are going to be pretty short, but they're important to know. And so we're going to go ahead and get started. First up, we have the karyotype, which is actually just showing the genome of an organism kind of laid out. So all of their chromosomes in a 2D format so that scientists can get different views of what they can see. So a karyotype is known to describe the amount of chromosome count for an organism. In general purposes, could be like studying prenatal issues, looking at the chromosomes, seeing if there's any tumors. But it allows us to get a good idea of what genes go where within different organisms. The typical human karyotype, we already know, consists of 23 chromosomes, 22 of which are called autosomal. Autosomal chromosomes are any other than the sex chromosomes. And then we have one pair of sex chromosomes that dictate gender and gender traits, XY for males and XX for females, which we can see here. So XY for males, and we have XX for females. And so those are called the sex chromosomes. So other than that, we have 22 autosomal chromosomes and one pair of sex chromosomes. But that's the karyotype. Next up, we have mutations. Now, mutations are going to apply to basically anytime we have DNA replication, mutations can occur. All they are is just errors in the DNA during replication. Most of the time, these errors cannot be passed on. If they happen in somatic cells, they cannot be passed on to another organism. The only time they're going to be passed on is if they happen in the gametes. If they happen in the gametes, it's called a germline mutation, and it can be passed on to offspring. And the crazy thing is, if you have a mutation in the genes of a zygote, for example, every cell after that is going to have the same mutation. And so it's kind of cool that that will continue to replicate with that error. Whereas if it happens in a body cell, it's just going to be that one cell that replicates and continues to have that error. So it's kind of like a lesser deal. And so it also doesn't get passed on to other children. So germline mutations in the sex cells do get passed on. Otherwise, the mutation is going to be localized to the organism and not to all organisms. But we're going to talk about the different types of mutations. For the most part, that all has to do with errors as the DNA is going to be duplicating or replicating itself. There's a category called point mutations, and then there's also going to be frame mutations that we're going to talk about. Frame shift mutation, we're going to talk about it a little bit later. But here's the general idea, in which you're just going to see like the genes are just going to have something happen to it. And it doesn't have to be a full gene. It could just be a single nucleotide, as you see up here. Like deletion is, in this example, deletion, they just delete one single nucleotide, or substitution, where they substitute this single nucleotide. Either of those can create dramatic shifts. And the reason why these mutations are a big deal is we already know that the DNA is like an instructions manual. So it acts like a book. And if you could imagine where you had a book and you tried to read that book, and then all of a sudden there was an entire page missing, well, now that book doesn't make any sense and you'd be very confused. And so is the cell. So if the cell reads a portion of DNA and now it has a duplicate page, or it has a missing page, or a new page inserted in, it's going to be very confused and it no longer makes sense. And so it's not going to build the protein correctly, or maybe it builds a protein that doesn't work at all. So all these issues can happen, and that's why mutations are a big deal. Now, not all mutations are a big deal. Some of them can just happen and nothing actually happens to the organism. But there's also going to be ones that can be detrimental to an organism. And there's also some that are beneficial. So a lot of mutations have a wide variety of things that can happen. So first, we're going to start off with point mutations. Point mutations are mutations that apply to a single nucleotide. So there is going to be substitution, as we talked about before, where you just substitute a with a T, for example. And it's just going to make a very small thing. It's not a big deal. Point mutations are generally less impactful because you're only switching single nucleotides. You could also have deletion, where you delete that single nucleotide. Um, the general idea here is it's just a small error, and your body can generally kind of fix it if it catches it early enough. So it's not a big deal. Um, there are also two different kinds of point mutations, called the missense and the nonsense mutation. When we talk about a missense mutation, that results in different amino acids being encoded because the positioning of the mutation is in the wrong spot. And so it kind of codes for a different protein than it intended to. And then a nonsense mutation is one, and because of that point mutation, it kind of stops the amino acid chain early, producing a stop codon, and then it just doesn't make any sense. It's way shorter than it was supposed to be, so it's just non-functional. So missense is like, it made a mistake in the spelling, kind of, and it made an error in the protein, whereas a nonsense will end it early by accidentally producing a stop codon and causing the polypeptide chain to just be way shorter than it's supposed to be, and it doesn't even work. So those are two things that can happen from a point mutation, either a missense or a nonsense mutation. And we can see here we have the final one, which is called a frame shift mutation. Now, frame shift mutations are way more drastic because they involve entire portions of the genetic code. It could be portions of a chromosome, it could be multiple nucleotides, or it could be a full gene that is accidentally mistaken. And so it has a big impact because obviously you're mistaking a larger bit of the genetic code in this case. And so you got a full protein that's supposed to look like this, but you only end up with this in the end because of the mistakes of a frame shift mutation. So these are a bigger deal than the point mutation would be. So our first frame shift mutation will be called duplication. And duplication is where a portion of the genetic code is literally copied twice instead of copying once. And so with duplication, you have some spare genetic material that is around. And actually, the cool thing about duplication is that it's actually an important phenomena that happens pretty commonly in organisms, and very often in plants, for example. When this happens, it actually kind of frees up an extra set of your genes to occupy a new purpose or a new role. And so, if these mutations occur, it can allow the species to evolve a lot easier because it has these freed up genes to take on a new purpose, to evolve a new limb, or to create a new set of eyes that weren't previously there. And so duplication has a great purpose within evolution. However, it can cause issues if unchecked, depending on what mutation and what genes are actually going to be duplicated. So duplication, very, very interesting. It has great positives and also tremendous negatives. Next up, we have deletion. There are two different types of deletion. We already talked about the point deletion, where you just delete a single nucleotide. But there's also segmented deletion, which is called macro deletion, where you delete an entire section of genetic code, and it results in the chromosome being smaller than it's supposed to. Obviously, like, this is like having that missing page in the book. It can create a whole new protein that's not supposed to be like how it's supposed to be, or that protein just doesn't work at all and it's non-functional. And that's obviously going to create some issues as well. Um, some diseases that can come from deletion are like cystic fibrosis, which is a life-threatening mucus that blocks the lungs and the airway and can cause you to get lung infection really easy. And basically, you're always going to be on some kind of something, some kind of life support because of cystic fibrosis. So definitely deletion is pretty much more harmful than not most of the time. Then we have insertion, which you have, again, the point insertion where you can just insert a pair of nucleotides to the DNA. Then you have macro insertion where you can insert an entire segment of genetic information into the uh, genetic code. And it's usually going to come from another chromosome. It's just going to be inserting one piece of chromosome to another chromosome. So it's the same genetic information. It's just going to be inserted somewhere else, kind of like shifted over. But because, again, if you think of it like taking a section of pages from one book and putting it in another, it doesn't matter that it's the same genetic code. Technically, it's in the wrong spot. And so that book no longer makes sense. And that's also a problem. So you imagine all of these are very problematic. Now, the problem with mutations is that they can also cause things like cancer. Cancer being one of the major causes of cancer being these mutations. Um, the It's a good thing that although this actually happens a lot and cancer can become a result of it, your body's actually very good at dealing with these mutations and keeping you healthy. And so in most cases, when there are cancer cells, your body will actually kill them. It only happens once in a while that there are cancer cells that continue to propagate with your body not noticing it going unchecked. Other ways that you can get mutations could be from chemicals, exposure to too intense chemicals. We know that radiation, like UV radiation, for example, X-ray radiation, those higher intensities of wavelength from the EM spectrum can cause cancer, and also viruses. So viruses actually cannot reproduce on their own via asexual reproduction. And so what they actually do, if we go back one, is they will insert a portion of their genetic code into your cells, and then your cells will learn how to replicate for the virus. So they use your cells replication function in order to create copies. Then they burst out of the cell and spread to other cells. And that's why viruses are such a big deal, because they insert their DNA into your genes and ask your cells to replicate for them. And that's we just talked about the problems with insertion, so it can cause cancer. And so yeah, it can also cause these mutations because of the changes. But that's the end of mutation. That's the end of karyotyping. I hope you guys learned a lot from this video. Hope send an email, answer the questions, and I'll see you later. Bye.